Morning, guys. Can you hear me?
Morning, doctor. Hey, you.
Okay. Sorry. Ngantuk ni. Ya Allah. Jangan ni Jumaat. Esok saya kerja juga ni. Okay. Before I start, good morning. Assalamualaikum. Salam Jumaat. And do you have any question before I begin? From previous class, anything, your assignments or whatnot. I haven't checked the class today. Wait. Ramai lagi tak post dalam Padlet ni. Saya bagi markah kan. Okay. For your assignment, your class course rep asked me before whether it's a group assignment or it's an individual assignment. It's an individual assignment. It's an individual assignment. So each one of you have to prepare it. So you basically those listed are important diseases, important topics. So please, it's a good chance for you to study lah. Okay. And for today, while waiting, wait, how many people here today? 84. So there's still some. Okay, while waiting, so let's have a short this kind of thing. Just asking you a question related to the previous class. Okay, this one. Over the past 2 weeks, last week you have learned about the diseases during the brooding period. Okay, so this is a question for you. Over the past 2 weeks, early chick mortality has been increasing in 2 of an integrated broiler company farms. The following are mortality charts from representative houses. Farm A, this is the mortality. Farm B, that is the mortality pattern. So the question is, list 3 the predisposing factor leading to the mortality pattern in Farm A. List 3 specific causes of mortality pattern in Farm B. Okay, anybody want to try on this? Hello? Anybody? Or you want me to call names? Or you want me to give marks?
Hello, doctor.
Yes?
I think for Farm A is
I don't think
during the
that. Just wondering who's that. Is
Iskandar.
okay.
I think for the Farm A is during the transportation.
Mm-hmm. And then Farm B?
Farm B is the farm management because on day one Farm B has low mortality rate.
Okay, that's correct. Okay, so basically, in the difference in between Farm B, you see that in Farm A and Farm B, okay, the mortality increases within the first few days. Okay, it's peak during the first few days and it's decreasing over time. And then in Farm B, it starts off okay, and then over time the mortality increases. So the problem should be the Farm A. Okay, the Farm A, most likely the issue is related before the chicken even arrive at the farm. Okay, transportation stress, vaccination handling, low quality chicks, aspergillosis or anything yang apa tu? Yang dia kata let's say the moment you put the chicken there, those that weak are just dying, and then those that survive will survive over time. So it's not an increasing mortality. The mortality will peak the first few days because of the bad quality of the chicks. When they arrive, they just almost die already, and then the one that died will die, and then the rest will survive. In comparison with Farm B, the chicks arrive okay. Okay, the chick arrive okay, but there's something wrong at the farm. Okay, maybe the farm is not hygiene. It's very dirty. They get infected with E. coli. They get infected with aspergillosis, or their brooding is not good. Okay, the brooding management. The temperature is too high. the ventilation is not good, and then the farmer has been rough, the food given was insufficient, and then you can see an increasing mortality. So that's why A, they call it predisposing factor. There's a lot of factors can contribute to it. But for B, specific causes, because usually you can determine what is the problem. Okay, so that's the answer for this. Okay, good job. Okay, for today, we are going to cover on respiratory diseases, and I'm just going to cover Newcastle disease. I haven't uploaded the notes yet, okay, because I'm trying to sort of like modify the notes and simplify it. It's a bit too much. I want to update a lot of things. I will upload it soon, okay? Because now I will give you presentation from mine, and then there's a slide presentation was given by the VM5 before. I asked them to do a revision class for their class. Okay, they are doing a pretty good job. Okay, they collected samples and whatnot. So I will present it on both sides of this to you. Okay, so let's see. Okay, Newcastle, first of all, respiratory diseases. Respiratory diseases, when it comes to diseases in poultry, is the most common disease. Is the most common disease encountered by poultry, because that's the most common disease that easily spread. Okay, they're easily noticed. Okay, you will see they're showing clinical signs quite easy. Okay, they usually the mortality also varies, but most of the time it's quite high. Okay, these are the important diseases, respiratory related diseases when it comes to poultry. For bacteria, infectious coryza, colibacillosis, mycoplasmosis, fall cholera, virus, we have Newcastle disease, avian influenza, infectious bronchitis, infectious laryngotracheitis, fowl pox. We also have fungus, aspergillosis, we have parasites like Syngamos trachea, and others could be related to management problems such as pneumonia. Before we proceed further, okay, one important thing that you have to know, you can see my screen, okay, is you have to understand the anatomy of the respiratory system. Please revise back your anatomy. So in poultry, in avian, okay, the difference is the avian, the lung is like a solid object. It's not like mammals where you expand and inflate and deflate together with the diaphragm. The chicken does not have diaphragm. The respiratory system is slightly different. They are unilateral flow. Okay, they flow one way, and then they use the air sac for the airflow. Okay, they have to expand the air sac for it to airflow. So in any part of the organ, okay, as whether the air sac, the lung, or the bronchioles, or the trachea is affected, the chicken will show respiratory signs. Okay, that's why even the chicken have air sacculitis. Air sacculitis is like thickening of the air sacs, and then the air sac is not elastic as previously. It's supposed to be quite elastic lah, expand and deflate. Okay, bila dia jadi thicken or have caseous material, they are unable to inflate and deflate normally. So they impair the respiratory function of the birds. Okay, so please revise this back. Okay, the first disease that I'm going to talk about today is Newcastle disease. It's one of the most important disease, not just in Malaysia, in the world, and it's quite important at the moment because right now Malaysia is facing quite a number of Newcastle disease outbreak. Okay, it's like everywhere. It start off in northern area, I think, and then comes to central now, uh, affecting Johor and semua tu. Okay. So previously, it was known as avian paramyxovirus type 1. It was classified. Now it's classified as avian orthoabulavirus type 1. So this is the new name lah. Okay, the new name. So Newcastle disease virus they belong to different isolates. Okay, and the strains may produce enormous variation in the severity of the disease. Based on the clinical signs, there are 5 forms of the disease: Bish form, Dudet form, and then these forms are you don't have to memorize all these forms so much, but this one is the much more important one, the petotypes. Okay, they are classified into velogenic, mesogenic, lentogenic, and the hepatogenic petotypes. Let's go back to here. Okay, it's just introduction. Okay, Okay, this is the autoalveolar virus, virus. It's a single-stranded RNA enveloped virus. They belong to only one serotype and has been grouped into 5 petotypes on the basis. These are the 5 petotypes: viscerotropic velogenic, neurotropic velogenic, mesogenic, lentogenic, and subclinical one, more like hepatogenic. Okay. So viscerotropic velogenic is highly pathogenic form in which the hemorrhagic intestinal lesions are frequently seen, like we call it the Doyle form, like in my lecture notes just now. And we have neurotropic velogenic, a form that present with high mortality, usually following respiratory and nervous signs. Okay, we call it the Bish form. And we have the mesogenic, a form that present with respiratory signs, occasional nervous signs, but low mortality. It's not as bad as velogenic. Velogenic, you will see mortality can be as high as 100%. They just die. Okay. And then lentogenic or respiratory are so form that present with mild or subclinical respiratory infection. Usually, sometimes the chicken don't even show clinical signs. Okay, it's almost similar with the subclinical, a form that usually consists of subclinical enteric infections. The history of this is the first outbreak was reported in 1926 in Java, Indonesia. However, at the same time also, it was discovered in Newcastle upon Tyne, the name of the place in England. But since you know lah, these Western people, they will use the Western diseases, so we call it the Newcastle disease, or else we will have called the Java disease instead. Okay, and it was discovered in many other places as well. In Europe, 1926, in Korea, 1924, with similar clinical signs. And nowadays, distribution is worldwide. Okay, this like any poultry producing country, they must have reported Newcastle disease. Except in US, they do still have Newcastle disease. It's just that they don't have the velogenic Newcastle disease. They have mesogenic and lentogenic. Okay, so they call the velogenic one as exotic Newcastle disease. They have different names. If you read old papers, okay, old journals, you may have stumble upon a lot of names. Okay, pseudofowl pest, pseudovagal pest, pseudopoultry plague. So you don't have to memorize all this. There's few things that is important. For example, the Ranicat one still being used. The tello also commonly being used. Distemper sometimes. Okay. And then in Malaysia, what does it call in Malay? What do we call it? Any idea? Bahasa Melayu. Tampar ayam. Okay, ataupun kepala teling. Kalau any farmers, you go to the farm and then they say, oh, ni kena sampah ni atau kena kepala teling ni. So they are referring to Newcastle disease. Okay. So for this disease, the virus have some biological activities related to the activity. They have hemagglutination activity, which means that they can agglutinate red blood cells. Okay, due to the binding of HN protein to receptor of the surface of the RBC. So that's why for this virus, you can do HA and HI. They also have neuraminidase activity that acts as receptor site to help F protein to bring about fusion of the virus and cell membranes. We have cell fusion and hemolysis. Okay, and they are also quite resistant to several agents. Okay, infectivity may be destroyed by treatments such as heat, irradiation, oxidation process, pH effect, and chemical compounds. And then the rate of resistance depends on the strain of the virus, the length of time exposed. That's why if you want to disinfect farm, you need a lot of contact time. Issue before contact time, exposed with the materials, quantity of the virus. Okay, if the dose is too high, you may need more detergent. Okay, more disinfectant to denature it. Nature of the suspending medium as well. And then the strain can be classified into petotypes. Okay, based on the pathogenicity test. We do MDT. Okay, we call it mean death time in eggs. I think was okay. Yeah, petotyping of MDV can be done through intracerebral pathogenicity index and mean death time. Okay, or minimum lethal dose. Intracerebral pathogenicity index is done by basically you infected the chicken, 10-day-old chicks, okay, into the cerebrum, okay, and observe for clinical signs and mortality. And then you give score 0 healthy, 1 sick, and 2 dead. Velogenic will have a value approaching to 2 because all of them dead. Okay, lentogenic give value close to 0. So you have to put in 10 chickens, so you have to calculate the average of the scoring system. Okay, whether they are belong to lentogenic, velogenic, mesogenic. And then another one is we do based on mean death time. Is the highest virus dilution that cause death of all in the embryos. This one you inject in the eggs only. Okay, and then you observe within 90 hours. Okay, if they die less than 60 hours is velogenic, mesogenic 60 to 90 hours, and is less than that is more than 90 hours. So that's how we classify it. Okay, and then we also have genotypes. Okay, before I proceed further, because in this lecture I will talk about a lot about genotype, petotype, serotypes, all those things. Are you familiar with these terms? Have you heard of this before? Can someone define it? Anybody? Panggil nama ni. Okay, anybody? Want to try? What is petotype? Or what is genotype? Let me call name. Farman, are you there? Or you just turn on and then go to sleep?
Yes, doctor. Yeah.
Yes, tell me, what is petotype?
Petotype is organism that belong to the same species.
Not quite correct. Okay. Anybody wants to help Varman? How about serotype? You classify the organism, okay, based on petotype, serotype, and genotype. They are all of them have Newcastle disease virus, for example, but you classify them based on petotype, based on genotype, and based on the serotype. So what does it mean? Anybody have idea? Joey, are you there?
Yes, doctor.
Yes, answer me.
From the chat box, it says petotype is based on the degree of causing disease.
Yes. Okay. Petotype is defined as based on the pathogenicity. Okay. How severe it can cause the disease. Okay. A lot of diseases are differentiated into different petotype. For example, in Newcastle disease, they can divide it into velogenic, mesogenic, lentogenic. The velogenic one, they cause 100% mortality. Mesogenic, the form is like less. Or Marek disease can also divide it into petotypes. They have the virulent, very virulent plus, mild, and very virulent. Okay, so they classify based on pathogenicity. How about serotype? Clarence Lau, are you there?
Yes.
Okay, how about serotype? Are you familiar with serotype? You have heard about it before?
Can't really remember. Is it like there is certain variation for the virus, therefore they classify into a few serotype.
Yeah, and then, I mean, how they classify into serotype? What does it mean by different serotype?
Different serotype. Because there is some variation in terms of the genetics.
That one is genotype. Based on surface antigen, possible, but not quite. Any idea? Okay, let's put it this way. COVID virus. COVID virus, they put into Delta, Omicron, whatever name before this. There's a lot of fancy names. Okay, what are those? Are those genotype, serotype, or what? Or you didn't bother to even look at it?
Serotype.
Huh?
Serotype.
Serotype. Okay. Okay, serotype is a classification based on the sero. Sero is like serological test. Yes, like right? It's based on the antibody production. Okay, if the antigen is also related to the surface antigen as well. Okay, so in some diseases, okay, they only have one serotype. For example, one of it is Newcastle disease virus. It doesn't matter which strain, whether they are belogenic, they are mesogenic, they are lentogenic, they produce the same type of antibody that combat to fight against it. Okay, it's quite easy. Quite okay. Another the opposite is okay, like infectious bronchitis or COVID. Both of them are coronavirus. They can mutate quite fast, and then they can belong to so many serotypes. Each of them, each of the different serotype of this virus, okay, they can induce different immune response. So if the that's why for those that have different serotype, the vaccination strategy is gonna be difficult. Okay, if you for Newcastle disease virus, any vaccine will work against it because they produce the antibody that can fight against any strain. Okay, but those that have various serotypes, if you vaccine them using serotype A, okay, and then they get infected with serotype B, the antibody produced against the serotype A, the vaccine tu, they cannot protect against serotype B. They might have some cross protection. Okay, that's why they said previously they said the current vaccine may not be able to protect against the Delta virus. Okay, so that's why you have booster so many times. After so booster, then only can they protect. Okay, and then they don't care so much about Omicron. Omicron is a less virulent, so maybe they protect against it or something. So serotype is classification based on the production immune system, immune response towards it. And then the last one is genotype. What is genotype? Any idea? Hanif, are you there?
Yes, Doctor.
Yes, saja. What is
Genotype.
genotype?
Genotype is
Classification based on?
based on their genetic
Yes, the genetic sequence.
the
Okay,
DNA.
so they do a sequencing and then based on the genetic sequence also. So those are the genotype. So for Newcastle disease, they are unique. They can divide it into he's not divided into serotype because it only belong to one serotype, but they are divided into different petotypes and different genotype. Okay. So this is the NDV genotype. So they have quite a number of genotype. I don't think you need to memorize it. It's just that in Malaysia or in Asia, genotype 7 is the most common one. They have a new classification for it. I will put in the lecture notes. So you can read yourself. If I explain here, I don't think you will be able to understand as well. Previously, they have all this classification into genotype 7A, B, C. We have a lot of this genotype 7 in Malaysia. Nowadays, they classify into V.2, V.11, V.11, all those things. Okay, to simplify it. There is a journal on this. Okay, that you have to read it, but I don't think this is important for you at the moment. Okay, if you work in poultry, later on, this is important. You have to know all these things, especially if you are doing a lot of research related. Okay. Let me see. Next one is laboratory host system. Okay, the virus can infect animals in chickens and non-avian hosts, rabbits and mice as well. It's they call this Newcastle disease sort of zoonotic, but it's not really much of a concern. Okay, it can only cause conjunctivitis in human. Mata you akan jadi berair-air, megah-megah je. Okay, that's all. And only if you are sort of immunosuppressed. Okay, in normal human and anything, it's not you can't really infect humans so much. Okay, even if you infect you, it just cause conjunctivitis. Okay, laboratory and then the virus can also grow in chicken embryos as well as cell cultures. This is how we isolate the virus. Okay, NDV can replicate in enormous range of cells, but the best is CF. CF is chicken embryo fibroblast. Okay. The transmission of the virus is through inhalation, aerosol, ingestion, through fecal oral route. and mechanical formites, contaminated equipments, feed, footwear, and clothing. Okay, the incubation period of the virus is 2 to 15 days. The speed with signs appear depends on the infecting virus. If it's infected by velogenic, it can be as soon as 48 hours. You can see within 48 hours chicken dying like crazy. Okay, that's what I've seen in the past few months in a lot of farms. Host species, age of the host, the younger the host, the higher the mortality, the faster the incubation period. The immune status, if they are not vaccinated or that one, you will expect them 100% mortality. Infection with other organism, co-infection with HPAI or LPAI or immunosuppressive diseases will have much more severe signs. Environmental conditions in bad management, route of exposure, okay, fecal oral route usually the fastest one, and then the dose of the infecting. If they're infected with high dose of virus, then they will die a lot faster. The clinical signs you will observe is respiratory signs, enteric problems, and nervous signs. For respiratory signs, you'll see those with increased respiration, dyspnea, gasping, coughing, edema of the head, face on wattles, like all the face is swollen, eye discharges and nasal discharges, enteric problem, they're showing diarrhea, green watery diarrhea usually, okay? Nervous signs are muscular tremors, torticollis, paralysis, and opisthotonus. Okay, these are common neurological signs that you'll observe. In general, they just look weak. Okay, the uniformity is affected and they just die. Mortality can reach 100% in fully susceptible chicken. Those are not vaccinated or vaccination failure. This is an example of those farms with virulent Newcastle disease virus. I have tomorrow, one of your student, one of your senior will present one of the case. This is from the farm that I visited with the student last semester. Okay, the chick look small. They just look weak, cannot open their eyes. They're just sitting down. The face is swollen and red. Okay, and then this is the video. This is a torticollis of the chicken in the farm. The head is like twisted. Okay. Many of the chicken like that, and the rest of the chicken just sitting down, very weak. In this farm, their mortality was 50%. And then the population of this farm is 200,000. Can you imagine 100,000 chicken died? Where do you put all the dead chicken? Every day, all the farmer have to collect carcasses. Okay, like berguni-guni, but it's become a mountain of chicken carcasses. Okay, previously, if they have like dead chickens at the farm, okay, dead chicken at the farm, not so many, normal mortality, they will give it to catfish breeder, pengusaha ikan keli. Okay, dia untuk bagi makan kat ikan keli. But nowadays, kalau letak 100,000 ayam tu penuh, ayam lebih banyak daripada ikan keli kan. Okay, so that's what happened last time for virulent Newcastle disease. And one of the things you will see like reddening of the shank, okay, cyanosis of the comb, swollen of the head, and then eye discharges. So for the lesion, you expect to see conjunctivitis, tracheitis, asacchalitis. For the GIT, hemorrhages and necrotic or focal lesion in the mucosa of the intestine. And nervous system, you will not be able to see anything grossly. But if you take histology of the brain, okay, you will see neuronal degeneration and prevascular cuffing. I think you have learned this term in pathology. Okay, so this is how it looks like. So the carcass just look reddened. Okay, and then you'll see this is one of the typical common finding when it comes to Newcastle disease is hemorrhages of the proventriculus. Okay, this is the gizzard pedal. Okay, the gizzard and hemorrhages of the proventriculus. Again, hemorrhages of proventriculus, even from the case that we had last time. Okay, this one, hemorrhages of the proventriculus. And then this one is hemorrhages of the cecal tonsil. These are the few things that you look for. And then intestine, you will see patches of hemorrhages. They just reddened and congested everywhere. So basically, they just so this is the Peyer's patches. Okay, and then this, if there are older birds, okay, laying birds, you will see the ovary is also hemorrhagic. So this is some of the images. This one is due to IP. So basically, this is how it look like. They just congested and hemorrhagic in general. In some cases, you'll see some of these gaseous material surrounding the heart and the air sac. Okay, this how the, like I told you just now, if the air sac is affected, they become thickened like this. Okay, it won't be able to expand. Air sac is supposed to be very thin as thinner than paper. It should be like a piece of a layer of tissue. Okay, you should through and through. You should be able to see inside it. Okay, in this case, okay, it become gaseous material, and it impair the respiratory system. So for diagnosis can be made based on the history, signs, and the gross lesions. Like I showed you just now, they usually have some history of respiratory signs. Okay, they show signs, and they see the gross lesion just now, they just hemorrhagic everywhere, and then you look at the hemorrhages on certain places. And then you can do diagnosis based on the serology as well as PCR, okay, or isolation. Usually the most common one is serology and PCR lah. We can do HA and HI test or ELISA test. This one is either virus neutralization test, plague neutralization test, immunofluorescent antibody test, immunopraxidase test, and molecular test, which is the PCR. But the most common test is the serology, which is the HA, HNI, and also molecular test, which is the PCR. This is how they do lah. Okay, if I want to isolate in chickens. Okay, viral isolation by inoculation in the allantoc cavity of 10-day-old embryonated eggs. If you had taken virology, you should have done it during your practical class. And then you do hemagglutination inhibition, ELISA, or PCR. This is the most common test that you do to diagnose Newcastle disease outbreak. For control, we do have vaccination. Vaccination can trigger several immune systems, cell-mediated immunity, humoral immunity, and the local immunity. This one, I will refer to this one. Okay. Okay, let's go to vaccination. Two forms of vaccines commonly used, which is the live attenuated vaccine or the kill vaccine. It mimics natural infection, induced all the three responses, like I said just now. Cell-mediated immunity, humoral immunity, and the local immunity. Example of the vaccine strain are the mesogenic, Roakin, Komarov, Mukteshwar, You don't have to memorize everything, but it's good at least you remember one lah. At least Lasota. Lasota is the most commonly used. Okay, and we have the subclinical one. This is the strain of virus they use to make the vaccine. Okay, they also have other type of vaccine available. We call it recombinant vaccine. They use HVT virus, okay, of Turkey, which is the Marek disease, and they insert the F protein of the Newcastle disease into the virus, and then they give during day old. Or this HVT can also be given in eggs. Okay, at 18 days is of embryonation. In US, they do in eggs because that is a lot cheaper, because labor cost in US is much more expensive. So usually they vaccinate in eggs. But in Malaysia, we don't have in ovo vaccination. They do have one company used to have that, but maintenance of the machine is quite expensive, so they use labor on subcut. Okay, they also have the reverse genetic vaccine, genotype mesh live attenuated NDV vaccine, which is one is Prof. Aro currently doing it. Okay. Vaccination strategies protect against morbidity and mortality, but do not stop either the viral infection or viral excretion. They just stop against the clinical signs. A combination of live and inactivated ND vaccines at 1 day of old induces a higher and longer lasting humoral immunity when compared with an activated vaccine administered alone. Vaccination in boilers usually require 2 doses, but sometimes they just do 1 dose. For this one, I will show you this one. Okay. vaccination. We have a lot of vaccine strain, and different vaccine strain have different immunogenicity. Immunogenicity means like different strength of immune response. Okay, Mukteshwar strain will have a very good immune response. The antibody titer will be very high. But at the same time, all these good strain, okay, they can cause also bad post-vaccination reaction. To develop a vaccine is not easy. You have to balance between good immunity and the post-vaccination reaction. This one applies to all. When they develop the COVID vaccine also is the same thing. They want to find a vaccine that can give very good protection. If possible, the protection can across all strain, Delta, apa semua tu. At the same time, they don't want any post vaccine reactions. So the vaccine that you have, Pfizer lah, all those things, they have selected a lot of strains already, and then they probably have selected the best strain that give the less the good immunity, but with no post reaction vaccine reaction. Okay. So breeder they have quite a number of vaccination for it. They do it almost every month. Okay. But I'm not going to. For you, for breeder, you don't have to know lah. Breeder and layer. But for broiler, you need to know. Okay. There's few options of doing the vaccination. Number one, you do hatchery vaccination, give a day old, and then you booster it at second dose at 2 to 3 weeks, 14 days to 21 days. Okay, optional. First vaccination you give through spray or eye drop, and then second you give dry drop, drinking water or spray. Okay. And then they have another different vaccination strategy. Like they do 3 times. Number one, if within first week, they give the second one, and the third one is optional kalau nak bagi. Tapi sekarang ni, during this current outbreak, a lot of farms have done the 3 vaccination. Okay, 3 vaccination. Like even right now, I'm sharing you one of this one. Okay, I have one company try to approach me. They want to compare different vaccine. Okay, different vaccine strategies, because right now a lot of their farm having Newcastle disease outbreak. These are the vaccine strategy that they use at the moment. If they give this company vaccine, they give this company vaccine, they give this company vaccine. Some of the farm they use double vaccination or triple vaccination. Okay, because they are so scared of the losses. Because the losses against Newcastle disease is so high, some company went bankrupt. So they rather spend on vaccination, add some more, and then they earn probably less. Okay, they earn probably well, but they don't mind. So that's how bad is the Newcastle disease. Okay. Other than that, of course, you have to improve your biosecurity. Proper carcass disposal. Some virulent strain can survive in the carcass for weeks. Bird-proofing houses. Okay. Wild birds are reservoir for NDV as well. They do not have the disease. They don't get sick, but they can bring to your farm. Routine equipment disinfection, contaminated formats can become a source of transmission, and limitation of human and vehicle traffic during outbreak, especially. During an outbreak, you isolate and cull infected birds, thorough cleaning and disinfection of the farm using chlorine-based chemicals and lipids. Okay, ini vaccination. Eye drop ni IM. IM ni pun selalu kita bagi dekat dada. Tapi kalau untuk ayam kecil, dada dia kecil lagi, so you bagi dekat thigh pun tak. Okay. Yang ni pun ada banyak differences on the live inactivated and vector vaccine. You don't have to know all this. It's too advanced for you. Yang masuk exam kan nak baca semua. Student ni dia tak boleh bagi nota. Bagi nota dia nak hafal semua benda. Okay, so that's why I don't want that's why I told you I haven't uploaded the notes yet, because I want to screen only the important one. Previously I do give a lot of all this stuff, and then students tend to memorize it, which I don't like it. It's not important at all. Okay. I like to share because it's good, but somehow students don't know how to pick which one the important information. Okay, so like I said just now, paramyxovirus, there's a lot of serotypes of paramyxovirus. Okay, but Newcastle disease only belong to serotype 1. Okay, if it's serotype 2, serotype 3 or something, they cause different diseases. It's not Newcastle disease virus. Okay, just giving you general idea. Okay, and then this is how they do the pathogenicity index, MDT, ICPI. They also have IVPI. Okay, intravenous pathogenicity index. Okay, this is how they classify into velogenic, mesogenic, and lentogenic based on this. Okay, ICPI and MDT. Okay, these are the example of vaccines, strains of viruses used as vaccine strain. Strain H, Mukteshwa, Komarov, Roikin, Lasota, F, Tichenor, V4. Okay, if you work in poultry, then you will be very familiar with this one. They talk like it's normal. Okay, oh, which vaccine do you use? Okay, this one. Okay. Okay, this one. Even UPM also have produced our own vaccine. We produce it with MVP, Malaysian vaccine pharmaceutical. Okay, previously. Okay. And I think that's all for Newcastle disease virus. It's an important disease. You must know it can cause high economic losses. Okay, that's why some of your senior presenting Newcastle disease almost every year. We encounter this kind of disease almost every year. So they will present this for their clinical conference. Okay, any question? Anything related to this or related to anything else? For avian influenza, tak cukup masa lah saya present next. Okay, if there is none, I think that's all for today. Okay, again, for those that came in late just now, for the assignment is an individual assignment, it's not a group assignment. And then try to and then introduce yourself in the Padlet as well. I'm thinking of putting on online quiz, but will give you some time for you to study. And I probably can give you some case study for you to self-study, get familiar with this kind of things. When is test one? I forgot already.
29th of April.
29 April.
Kalau ikut the course schedule.
Dah, yang tu macam dah nak raya dah. Raya bila? When is Hari Raya? Raya is the week before Hari Raya. Okay, please let me know if you want to have the test on 29 or you want to make it earlier. We can have on 25th, which is on Monday. Tak adalah, in case lah korang nak balik awal ke apa ke. Nak masak nasi, nak masak tupat lah pelbagai. Okay, so that's all. Just let me know lah for the test. We can do it earlier. Okay, anything else? If there is none, I think that's all for today. Thank you, thank you for listening. Salam Jumaat. Okay, dan selamat berpuasa. Have a good weekend, everyone. See you guys.
Thank you, Doctor.
Thank you, Doctor.
Mau buka, Doctor.
Selamat berbuka. Tak nak buka dah ni pun.